Previously we have looked at the classification and structures of monosaccharides. In this lecture we will study some important chemical reactions of monosaccharides. The first type of reaction we will study is the formation of glycosidic bond. We have seen that monosaccharides undergo cyclization, and they have a free hydroxyl group on the carbonyl carbon, and they have a free hydroxyl group on the carbonyl carbon. The aldehyde or ketocarbons are also called as carbonyl carbons. This free hydroxyl group of the carbonyl can undergo a condensation reaction to form an ether bond with other groups. In this figure you can see the structure of alpha-D-glucose. This is carbon 1, the aldehyde carbon. You can see that it has free hydroxyl group which projects below the plane of the ring, so this is alpha anomer of glucose. By means of this hydroxyl, this glucose molecule can form a bond with other groups or molecules through condensation. We will now see how this happens. So let us first see the structure of alpha-D-glucose. Carbon 5 is linked to carbon 1, the aldehyde carbon, through this oxygen bond. Let us now see the hydroxyl groups. Since this is alpha-glucose, so the hydroxyl of carbon 1 projects below the plane of the ring. Hydroxyls of carbon 2 and carbon 4 project to the right side of the carbon, but the hydroxyl of carbon 3 projects on the opposite side. This is represented in the structure drawn. Note that carbon 1 is hemiacetal. Let us put another molecule of alpha-D-glucose. Let us call them as glucose 1 and glucose 2. This is carbon 4 of the second glucose molecule containing a hydroxyl group on it. These two hydroxyls can undergo a condensation reaction with the elimination of water. So the two monosaccharides will be covalently attached to each other through the oxygen atom. Such a bond where individual monomers of sugars are attached to each other through condensation reaction between their hydroxyl groups is called as the glycosidic bond. Note that the carbon 1, which is the aldehyde carbon of glucose 1, has no longer a free hydroxyl group on it and now is an acetal. The aldehyde carbon of molecule 2 hemiacetal, with a free hydroxyl, and in turn is able to form a glycosidic bond with another sugar molecule. This resulting molecule is called as maltose. Glycosidic bonds are not only present in carbohydrates but are also found in nucleic acids, DNA and RNA. This is a nitrogenous base shown in this figure. The hydroxyl group of ribose sugar in RNA undergoes condensation with the hydrogen of nitrogenous base. Remember that when sugars form a condensation product through their hydroxyls, the bond is a glycosidic bond. So the base is attached to the sugar through a glycosidic bond. Since this glycosidic bond is formed using a nitrogen atom, so this is called as N-glycosidic bond. The second important type of reaction is the redox reaction. Monosaccharides contain an aldehyde or keto group on them. Monosaccharides contain an aldehyde or keto group on them. These group can undergo oxidation and can be converted into carboxyl group. This oxidation is mediated by an oxidizing agent. In turn, the oxidizing agent itself is reduced during the reaction. Such carbohydrate molecules having a free aldehyde or keto group on them which can undergo oxidation are called as reducing sugars. Because they can reduce an oxidizing agent, and during the process they become oxidized. So their aldehyde or keto turn into carboxyl. Remember that in cyclic structures the aldehyde carbon is hemiacetal and the keto carbon is hemiketal. This figure shows some important types of oxidized derivatives of monosaccharides. One type of oxidation products are those where the aldehyde carbon undergoes oxidation to carboxyl. These are generally called as aldonic acids. They are named after the originating monosaccharide. Gluconic acid from oxidation of glucose, galactonic acid by the oxidation of galactose, and so on. The other type of oxidation products are those in which the alcohol group of sugar oxidized to aldehyde and then to carboxyl. Such sugar derivatives are generally called as uronic acids. For example, when carbon 6 of glucose undergoes oxidation, it produces glucuronic acid. The other derivatives shown are galacturonic acid from galactose and mannuronic acid from mannose. These oxidized derivatives have various important physiological implications. For example, bilirubin is a product of heme degradation. Accumulation of bilirubin results in jaundice. It is filtered and excreted by kidneys. Bilirubin has low solubility in water. Glucuronic acid is a polar molecule. It is conjugated to bilirubin and increases its water solubility and hence its excretion. In this figure, you can see two glucuronic acids molecules attached to bilirubin. The next important type of derivatives are lactones. Lactones are cyclic esters. Esters are condensation products between acids and alcohols. The oxidized sugars have a carboxyl group. This carboxyl undergoes condensation with the hydroxyl to form a cyclic ester bond I. E. A lactone. Another type of monosaccharide derivatives is alcohol derivatives. Aldehyde or ketone, upon reduction, are converted to alcohols. This figure shows some common alcohol derivatives of monosaccharides. Ribitol is formed by the reduction of aldehyde group of ribose sugar. FAD and FMN are redox cofactors. Ribitol is one of the constituents of the cofactors FAD and FMN as shown in this figure. The part of the molecule shown here is ribitol. Xylitol is another reduced derivative. It is produced by the reduction of a pentose sugar xylose. Xylitol is used as a sweetener. Reduction of glyceraldehyde produces glycerol, which is an important metabolic intermediate. Another important derivative is a hexose. It is called as inositol. It is produced by the reduction of glucose through enzymatic reactions. Inositol is attached to the polar head groups of membrane phospholipids. It is a source of inositol triphosphate IP3, which is important for cell signaling. The next derivatives are deoxysugars. As the name says, these derivatives lack an oxygen. One of the common deoxysugar we know about is deoxyribose. It is a pentose sugar and is found in nucleotides of DNA. The structure is shown here. Other deoxysugars include rhamnose and fucose. Fucose is a deoxy form of galactose. It is found in the ABO blood group antigens present on the red blood cells. When an amino group is attached to a sugar, it is called as an amino sugar, alternatively known as sugar amines. The figure shows glucose. The hydroxyl group of glucose undergoes condensation with amino. Hence the amino is attached to glucose with the elimination of water. This derivative is glucosamine. In the next figure, galactosamine is shown. These derivatives are important as they are added to proteins and are needed for the physiological activity. The derivative shown here is N-acetylmuramic acid. This part shown is glucosamine. Acetyl group is attached to glucosamine. This makes N-acetylglucosamine. The part of the molecule shown here is lactic acid. When lactic acid is attached to this molecule, the resulting structure is N-acetylmuramic acid. This is one of the constituents of bacterial cell wall peptidoglycan.